How's it going? My name is Cody Arnold, and I'll be talking about our Model 6 immune response flowchart. So we'll start with the antigen interacting with our body. So the first thing that's going to happen is going to interact with our innate immune response, and this whole response can happen within minutes to hours. And as we know in our innate immune response, the first thing that antigen is going to interact with is our first line of defense, which is our skin, which is that hard external, well, not hard, but that external barrier, which is our epidermis and dermis. And within that, you have dendritic cells, which we'll talk about later, which their whole goal is to phagocytize these pathogens. Next, also in that first line of defense is the mucous membranes, which on the inside of them, you have the epithelium. Now, a lot of times that antigen can get to this part and the dendritic cells will phagocytize these pathogens, or via mucosal shedding of the epithelium, these cells will break down and get shedded and pushed away, so not able to attach and cause infection. Now that second line of defense within the innate immune response, you have phagocytes, which are macrophages, which break down the antigen. You have toll-like receptors, which are a type of antimicrobial chemical, and they recognize self and non-self chemicals, and those non-self chemicals they can kill, which are also considered PAMPs. Then you have fever and inflammation. And like, for example, fever, our body temp increases to ultimately kill that pathogen in an environment that it cannot survive in. Now, if that antigen gets past that innate immune response and now enters our body, then you have the adaptive immune response, which happens. And this whole adaptive immune response can happen from days to weeks. Now, within this adaptive immune response, you have B cells and T cells that are working together ultimately to create this immune response. And they are called effector cells, because in unison they cause this entire immune response. Now we'll start with B cells, which are part of the humoral response. This is as the antigen is in the body but outside of the cell. So once these B cells are inside of the lymphatic system, they encounter antigens via BCRs. And what BCRs are, they sit on the outside of these B cells on the surface, and they have millions of them on the surface of the B cells, and each one is antigen specific. So that means they are capable of recognizing one antigen, but there are millions of them, so they can recognize a bunch of antigens, so they are specific antibodies for the certain antigens. Now, once they recognize this antigen and bind to the antigen is then brought into and onto the cell surface via MH2 molecules. And we'll talk a little bit more about that and what happens, because once this happens, the dendritic cells presents these antigen fragments onto the MH2, which allows these helper T cells to recognize it, and now that B cell becomes antigen resistant. As the helper T cells sense the sensitive B cells and activating and binding the MHC, they release cytokines to cause rapid division of the B cells into memory cells or plasma cells. Now, at the same time that this is happening, T cells are doing the same thing. Now, beforehand, these B cells that you have are plasma B cell or memory B cell. Memory B cells are inactive until they are reactivated by a re-entrance of this antigen, which then they can then rapidly proliferate into plasma cells to kill the antigen. Or upon the initial response, these plasma cells will interact with the antigen to kill it. But at the same time that these B cells are dividing via cytokines communicating with them to cause the division, T cells are doing the same thing. So the antigen is now within the cell. Okay, and the cell-mediated response is going to take over. So what happens is, once antigens are broken down and engulfed into many fragments, antigen-presenting cells take pieces of these antigen fragments, and what they do is they cause them to attach to the MHC, okay? And once that happens is you have T cells are able to recognize the pathogen and activate and cause cytotoxic T cell clones to divide into either inactive memory cytotoxic T cells or activated cytotoxic T cells. Now, these cytotoxic T cells are able to recognize the infected cells via these MHC protein complexes that are on the surface of the infected cell. They provide a readout of the proteins within the cell. So once the APCs bring those fragments to the MHCs on the surface of the cell, the MHCs tell the cytotoxic T cells, hey, there is a pathogen within this cell. Now, once this happens, the cytokines are released by these helper T cells, which we talked about beforehand, to activate macrophages and killer cells to ultimately engulf and kill this entire infected cell. These cytotoxic T cells are also activated by the cytokines, as well as the plasma cells, to ultimately enhance the ability of the killer cells and macrophages to begin recognizing and killing these infected cells. So my sources are down at the bottom left. Thank you so much.